My name is Edgar van der Grift. This is Charlotte, and we are both trying to moderate this session. And I hope you forgive us if we mess it up, but we try not to. I saw in the program that the first presentation, very interesting one, is myself. So let me start. All right. There it is. I'd like to talk to you a little bit about a method that we developed in the Netherlands for including scientific evidence in guidelines or handbooks that we produce on road mitigation. So it very much is a kind of a follow-up of the story that Silvio just presented plenary. The reason why we started to develop this method was that we are working on a handbook, a new handbook in the Netherlands. The previous one was 8 years old and it was time to update it. It's published last year, you can find it online. And the new handbook is really different from the old one. There's a completely new roadmap in it, how to decide on mitigation and prevention measures. Guidelines, of course, are updated based on new knowledge. It's much more concise. It's not an extended cookbook. It doesn't say you step by step what you have to do, but it's more presenting possible solutions, recommendations. It's also a different layout. It's an online PDF with active links, so it's easy to scroll through. And last but not least, and that's the topic of my presentation today, it includes scientific evidence, or we try to include it where possible. So what of the guidelines, what of the recommendations we present in the handbook are indeed based on science? And we thought that is important because that will give us much more insight in what guidelines are really supported by science, are really proven, are supported by research, and what of our guidelines are merely a result of maybe our practical experience or our expert judgment, or maybe are just a copy-paste result, like was just presented by Silvio. One handbook is copying what is written in other handbooks, and nobody at the end knows anymore where it comes from. By doing this, we will also gain insight automatically in where and what are the knowledge gaps. So it will help us to make a targeted research agenda. And the ultimate goal, of course, was to help policymakers, road planners to make better decisions, to make better mitigation plans. And you have to get me right, expert judgment is not wrong. Or practical experience, it's really necessary. It's often the only thing we have. But if we use expert judgment, we have to be cautious. Like a wise man recently said, expert judgment should always be distrusted, including your own. I always also say, you know, put 10 experts in one room and you get 9 different opinions. Because one is maybe following his boss and all the others have their own opinions and they're all different. So we have to be careful in treating expert judgment. Back to the handbook, the scope in a nutshell. It's about reducing wildlife mortality and reducing the barrier effect. And we looked at scientific evidence in terms of the effectiveness of measures. What can we find on that? But also the use of crossing structures. As you probably know, we do a lot of monitoring studies worldwide that look into the use of crossing structures much more often than that we look into the effectiveness. So it would be a pity to leave out all that information. Our aim was to come up with a method that is transparent, unambiguous, should be able to, or it should be easy to replicate it in the future if we want to make updates of the handbook, and it should be simple. We were thinking of an output like a traffic light. Red is obviously something that should stop you. You should not do that, you should not go there. And green would mean, yes, go on, step on the gas. This is a measure you can apply for this or that species. This was not easy, because you know there are many studies and they all differ in their approach, in their methods. They are different in their quality and sometimes even in their outcome. So one study may say, yes, this measure is great for a certain species group, and others might say, no, it's not so great. Actually, we found it didn't work. And we wanted to try to take all this into account. And the method we came up with was consisting of 6 steps. Here they are. First step is an easy one. We defined our species groups, our types of measures, and our crossing structures, so we knew what we were talking about. In the second step, we defined response classes for the effectiveness studies and use classes for the crossing structure studies. And after that, we followed 4 steps, step 3 to 6, for each type of measure or each type of crossing structure. First, we assessed the number of data sets that we could find. And we talk about data sets in this case because one study may provide multiple data sets. For example, if a study is about multiple species. In the next step, we assessed the quality of these data sets, which is very important. We used a set of criteria for that, and the set of criteria was different for the effectiveness studies and the crossing structure studies. And the criteria were all related, as you can imagine, to the setup, the study design of the work. In step 5, we assessed the scientific quality class for each response class or use class and for each species group. And we promised to keep it simple, so we distinguished only 3 classes: weak, moderate, and strong scientific support. And the last step is the transformation into a suitability ranking, and there we distinguished 4 classes from not suitable to highly suitable. Well, the result of step 1 to 5 is a set of matrices, and this is one of them, one example. This one is about the effectiveness of wildlife reflectors in reducing roadkill. You see the response classes that we distinguished, 8 classes in this case, from an increase in mortality to over 75% reduction in mortality. And you see on the left the species groups for which we found data sets, in this case 2 species groups. The numbers in the cells reflect the number of data sets, and the colors reflect the scientific quality of these data sets. So for example, for the medium-sized mammals, we found 4 data sets that we could use. They all are concluding that there is no reduction in mortality, and the scientific quality of these studies, on average, is strong. Interesting, provides a lot of information, these matrices, but for our handbook, our guideline, still too complicated. It's still not the traffic light we were looking for. So that's why we did the next step, step number 6, where we assigned a suitability class to each response class. So we simplified the situation. For that, we first had to calculate a suitability index. We called it G, and G was based on both the number of data sets and the quality of these data sets. And then we looked at which cell had the highest G value, and that one, for example, for large mammals, G value of 0.84 is the highest in the row. That made us decide, that made us decide that in this case, large mammals, reflectors, are judged as not suitable. And that we did for all the measures. And that brought us to the traffic light we wanted to achieve. These are the final legends that you can find in that handbook. First, a few general units about no research or insufficient research, but then the 4 colors that tell us the judgment about certain measures of or crossing structures. Output in the handbook looks like this. This is one of the tables. This is about the crossing structures. You see the species groups on the top. You see the wildlife measures, the crossing structure types on the left. And you see immediately an overview of the evaluation in the table. Is this the end of the story? Now the handbook is published. I think not, and I hope not. Of course, we made a selection of studies that we could include. We can add existing studies if we want, but more important, we should start new studies. We should start filling the gaps that became clear by doing this exercise. And if we do so, we can probably in the future also make similar analysis for other features, other design features, for example, dimensions of structures, or we cannot talk about species groups, but really individual species. So I hope to continue this work. I hope you will collaborate in this, and hopefully we are ready by 2045. Thank you very much. All right, questions, discussions, anybody? Sylvie.
Just in relation to the species-specific differences. So bats for me is a really good example, but I think we have similar situations with reptiles where in effect the grouping of all bat species in a bat category can be a bit misleading because, for instance, when you're looking at underpass usage, especially if there's something like a stream there, you can see that actually species that actually follow water features such as network bats overwhelmingly use the structures, but almost none of the other bat species present there use it. So it's really functionally specific rather than actually broader group. And I think certainly the data that we have for reptiles suggests a similar trend, where out of, I don't know, 5 species of reptiles in one site, one might actually massively benefit from the structure, the others a bit moderate, and one not at all. So I was just wondering whether there's a sort of simple solution instead of sort of aggregating the data like that. Thank you very much.
Yeah, it's a good point, and we only aggregated or clustered the species into groups because there are not enough data sets to split the species. So then you end up with a lot of situations where you have one species and one data set. So actually it's a lack of studies that made us make the decision like we have to group. But while interpreting or looking at the outcome, you should always keep in mind, yeah, this is about all large mammals, but we know they are different in their biology and their behavior. So it's always good to look a step further, but right now in Europe we cannot go very much further. A few species you can do that. A few species are well studied, mostly because they are important from a point of view of traffic safety. Then multiple studies appear. If you come to other species groups, smaller species, usually it really, really becomes rare what you find. So yes, you're completely right, and that's the future. We should make such a thing for each individual species, but right now it's almost impossible. And there are many species, nothing has been studied. Nothing. All right.